to give this tutorial on Benchtop NMR and how we're using parahydrogen hyperpolarization to boost the sensitivity of Benchtop NMR. So just before I start, I wanted to just say a few acknowledgements. So all of the work that I'm going to be talking about today has, of course, been done by my group over a number of years and with key contributions from various former students, current students, and postdocs. And so this is just a summary of some of the key people who did the work. And in particular, I'll be talking a lot about the work of Alistair Robinson, a former PhD student, and current PhD student, Anna Silva-Terra. So one of my main research interests is about thinking about what we can do with compact NMR instrumentation. And so if you think about NMR spectroscopy and how it's developed over the last few decades, there's been a very understandable push to work towards higher and higher magnetic fields using large superconducting magnets in order to maximize both the NMR sensitivity and the resolution in terms of the chemical shift separation. But of course, by moving to these very high field instruments, we are sacrificing portability and affordability. So this sort of cartoon here shows an example of three different NMR instruments that we have here at York. So we have a 700 megahertz spectrometer, a 300 megahertz in our teaching labs, and then this is sort of the dimensions of one of our benchtop NMR spectrometers that operates at 1 Tesla or 43 megahertz. And so the real benefits of the benchtop NMR spectrometer is that we don't need to put it into a specialized lab. We don't need to fill it with cryogens. So it's much cheaper to buy in the first place, but also more importantly, in some ways these days, is that it doesn't require a lot of maintenance and a lot of continuing costs once you've had the initial cost of buying the instrument. But of course, we have the difficulty if you're trying to use a benchtop NMR to provide access to the wealth of information that we know NMR gives us access to, that we need to battle the issues of sensitivity and chemical shift resolution. And when we focus in on the issue of sensitivity, then we can start thinking about ways around that using hyperpolarization. So just sort of a reminder of what we're talking about when we're talking about polarization is that our NMR signal comes from this very small population difference between our two Zeeman energy levels. So if you think of a proton, we've got spin up and spin down spin states. And the difference in population of the proton populations is at room temperature about 3.5 ppm per Tesla that you apply. And so that gives us a very weak signal, but a very, very powerful signal. But if you're working at lower magnetic fields, obviously you have a sensitivity issue. And so the idea of hyperpolarization is to force your system out of the thermal equilibrium into a state that's much more ordered, where we have a higher population of one of these energy levels. And this gives us opportunity to boost our signals by many orders of magnitude. So from tens to up to thousands or even tens of thousands. And so if we want to use hyperpolarization to boost the sensitivity of benchtop NMR, then we need to think about what technology we're going to use and how that fits in with the general benefits of benchtop NMR, which is that it's relatively compact and portable and low cost. And so my group is particularly interested in parahydrogen-induced polarization as a way of overcoming the sensitivity challenge of the benchtop NMR. So in this tutorial, I want to spend a bit of time just telling you a little bit about what parahydrogen is and how that gives us hyperpolarization, and then how we can couple that to our benchtop NMR spectrometer. So first, just from a bit of a historical perspective, parahydrogen hyperpolarization is one of the more recent hyperpolarization methods. It was first developed in the 1980s, and really, there was pioneering work on this from Bowers and Weidekamp in 1986, where they put out this theory paper predicting that you could use this special form of hydrogen to generate hyperpolarization, followed up by an experimental demonstration of the same effect the following year. Now, concurrently in the group of Rich Eisenberg, that what they were looking at hydrogenation reactions, and they were seeing unique behaviors that at first they didn't really understand. And so what they were seeing is that when they were carrying out these reactions with hydrogen, that if they had stored their sample overnight in liquid nitrogen, so this is common practice in order to stop any reactivity from happening until the moment when it was going to be measured, that then when they subsequently did their hydrogenation reaction, they got these enhanced peaks that had this weird antiphase behavior. And they were able to attribute this effect to parahydrogen through understanding the work of Bowers and Wedekamp. And so this just is a sort of interesting illustration of where a lot of this technique comes from. And just from a point of view of nomenclature, what you'll notice here is in this paper, this original paper by Bowers and Wedekamp, we have the acronym Pasadena. And so this is a common term used for a specific type of parahydrogen experiment that I'll discuss a bit later. And in this paper by Rich Heisenberg, you get the PHIP or PHIP acronym. And that's just because of its simplicity, is one of the general acronyms that is essentially used to talk about parahydrogen. Any hyperpolarization using parahydrogen would be referred to as PHIP. Okay, so from a fairly fundamental perspective, what is parahydrogen? So parahydrogen is a spin isomer of H2, of the diatomic molecule H2. So why do we get this particular para state and then the complementary ortho state in dihydrogen? Well, if you think about a diatomic molecule, we can break down its Hamiltonian in terms of translational, rotational, and vibrational parts, motional parts. Then there's the electronic part and the nuclear spin part. And so if you think about the weight overriding wave function, it's going to be the product of these various components. And the overall wave function has to be antisymmetric with respect to exchanging the nuclei. And so we can look at these different components. We can see that the translation, the vibration, and the electronic are all symmetric relative to swapping the two nuclei. And so we need to have this anti-symmetric behavior coming from either the rotational part, which is symmetric or antisymmetric, or the nuclear part. So what does the nuclear spin look like for H2? Well, if you think about H2, we basically have two protons, two spin half nuclei, but they're completely indistinguishable because it's because we can't tell the difference between the two hydrogens in H2. And so instead of thinking about them separately in terms of whether they're spin up or spin down, we need to combine their angular momenta, and they either combine. Sorry, I've lost track of what's on my slide. So basically, if you have two coupled nuclei, you look that up in the textbook, you might see a set of energy levels that look like this, where we talk about the spin up and spin down of the different nuclei. But this basically only applies when you have two weakly coupled spins where they have a different chemical shift and then a small J coupling. But this is not our case here because our two protons are completely equivalent. And so we need to couple their angular momenta together to form either a spin 1 state, which is a triplet state, ortho, and it's called orthohydrogen. And so the three states that make up the triplet are M equals 1, which is with spin up, up, M equals 0, which is a combination of the two forms of up downs, where the two are canceling out to give M equals 0, and then the down down state, which is the M equals -1. Now, our angular momenta can also combine to give us, to basically cancel out the total angular momentum. So we have total spin 0, and this is the parahydrogen singlet state. And so this is associated with the single energy level, which is our singlet state, which is the combination of alpha, beta, beta, alpha, so our two up down states, but now with a minus sign in between. So overall, M equals 0. And if we look at the exchange of nuclei in these spin states, we can tell that the orthohydrogen triplet state is symmetric. It's unchanged if you swap the two nuclei, whereas the parahydrogen is antisymmetric. So if we swap the two nuclei, we get a negative sign out. So they're antisymmetric. So if we go back to our overall wave function, we can see that both the rotational and the nuclear part can be symmetric or antisymmetric. So if our overall wave function needs to be antisymmetric, then we have a coupling between the rotational and the nuclear. So you can either have antisymmetric on the rotational or the nuclear. So we have this coupling that means that now the difference in energy between our para state and our ortho state is not determined by the Zeeman interaction, but by a much larger gap between the rotational states. And this is because our symmetric orthohydrogen state is coupled to the antisymmetric odd rotational states, and the antisymmetric parahydrogen state is coupled to the even rotational state. So our difference in energy is to do with the 0 to 1 transition in the rotational states. So what does that really mean practically? Why is that important? So what it means is that we have this large energy gap, which means that as a function of temperature, we're going to have different proportions of parahydrogen and orthohydrogen at different temperatures at thermal equilibrium. And if you think about the high temperature limit, so at room temperature, basically all of our energy levels have enough energy, thermal energy, to be roughly equally populated. So we get 75% orthohydrogen, 25% parahydrogen. And if you cool all the way down to 28 Kelvin, you get about 98% parahydrogen. And at liquid nitrogen temperatures, which is quite convenient to use, we get about 52% parahydrogen. Now, so this implies that all we need to do is cool down our hydrogen in order to overpopulate or to increase the population of the para state. But the thing to keep in mind is that transitions between para and ortho are symmetry forbidden, so they have very low probability. So this conversion rate is going to be very small. And we can use that to our benefit, because when we generate a hydrogen gas that's overpopulated in the para state, then we can maintain that for a long period of time, because the rate of relaxation back to orthohydrogen is very slow. So how do we actually do that? Well, in order to increase the proportion of parahydrogen, we just cool down our hydrogen gas. So we flow it through a coil sitting inside of, say, a cooling block, or sitting inside a bath of liquid nitrogen. And inside this coil, we put some kind of paramagnetic surface, so something like iron oxide. And when the hydrogen comes into contact with this paramagnetic surface, it breaks the symmetry between the two nuclei and allows for rapid conversion between ortho and para, and that allows it to come to thermal equilibrium at that temperature. And so what we do is we flow the hydrogen through, it comes into equilibrium at that temperature, enriches in the para state, and then we heat it up again, but now we make sure that we're not exposing it to anything paramagnetic. So we become locked in the para state. So we overpopulate the para, and we get our parahydrogen enriched gas. And if we're careful about how we store it, then it'll stay in that para state for potentially weeks or months. So just how do you do that from an instrumentation point of view? So the simplest apparatus to use is just based around liquid nitrogen. So this is a sort of home-built parahydrogen rig from my lab, where we have a large store of liquid nitrogen for the conversion. And that gives us, like I said, 52% enrichment. Over in the Center for Hyperpolarization, which is where my colleague Simon Duckett works, who has been a pioneer in parahydrogen hyperpolarization for a long time. They have a lot of home-built systems that work at 28 Kelvin, so you get near 100% parahydrogen enrichment. And then there's also a range of commercially available equipment, such as this generator from Brooker operating at 38 Kelvin. And so the idea is that you can basically create a flow of parahydrogen enriched gas to be used on demand. And while in principle you can store it for long periods of time, from a health and safety perspective, we would normally just use it in flow mode, or fill up a small amount in a sample, so that we're not handling large volumes of hydrogen gas. So for example, here you can see this is just a hydrogen generator that's just an electrolysis cell, so generating hydrogen from water. So just to sort of reiterate the point about the lifetime of parahydrogen, so once we've enriched our gas in parahydrogen, then in the gas phase, if you isolate it from any paramagnetic surfaces, it can last for weeks or months. If you dissolve it in solution, for example, these are a series of experiments in toluene at 500 megahertz, you can see that the back conversion from para back to ortho is very slow and happens over many hours. So it's a very convenient source of hyperpolarization, because it's very stable once it's created. But in a sense, it's sort of feels like we're going backwards, because if we're looking at parahydrogen has no overall spin, it's spin 0, so therefore it gives no NMR signal. And so in fact, what these measurements are doing are looking at the hydrogen peak in toluene and watching the hydrogen peak grow as the parahydrogen that's invisible turns into orthohydrogen that's visible. And so it seems counterintuitive that we're going to get hyperpolarization from something that doesn't give any NMR signal. But the point is that it's a very ordered state, so it has a high degree of spin order. So it gives us a very, very ordered state to start with. And then if we use it in a chemical reaction, we can then transfer that spin order into state that we can directly observe. And this is what gives us hyperpolarization. So how does this work? So here are the states that I've introduced before. So our singlet state and our various triplet states. And if we enrich our gas 100% in parahydrogen form, then we would only populate this singlet state. So now you imagine taking that feedstock, essentially, of parahydrogen gas, and we do a hydrogenation reaction. So say we have a triple bond that we then add the parahydrogen. So we hydrogenate the bond. So now we have 2 protons in our product molecule that have come from parahydrogen. So they started out in the singlet state, but now they have different chemical shifts if these 2 R groups are different. And so their symmetry is broken. And so what happens is if we carry out such a reaction in the magnet, so in the high field, so this is the so-called Pasadena approach, then the 2 protons in the product molecule creates our AX weakly coupled spin system, so we can use our standard sort of energy level diagram. And what happens is the singlet state overpopulates these alpha beta and beta alpha states. And so we get enhanced either absorption or emission transitions here that show up as these sort of antiphase doublets in our spectrum. And because we're starting from this very, very ordered initial state, we get very, very high levels of signal enhancement. The other way to do this experiment is to do it outside of the magnet, so in a much weaker field where your difference in chemical shift is much more similar to your J coupling. So you have a very, very strongly coupled sort of AB type system. And in that case, you're going to overpopulate preferentially one of these alpha beta or beta alpha, depending on the exact spin system. And so we still get these enhanced transitions, but you get a slightly different looking spectrum where we've only enhanced 1/2 of each of the doublet from up here. And often you would see some mixture of these two effects. So by carrying out a hydrogenation reaction with parahydrogen, we can see these remarkable levels of hyperpolarization. And this is what they were seeing in the paper I showed earlier by accident. They accidentally created parahydrogen by storing their samples under hydrogen in liquid nitrogen. So they cooled it down and created parahydrogen. And then when they did the hydrogenation reaction, they got these unique signatures. So how can we apply this on a benchtop NMR spectrometer? So what we were interested in this work, which was to look to see whether we can actually do quantitative reaction monitoring using parahydrogen hyperpolarization. So we chose a model system. So this is a very well-studied iridium complex called Vasquez complex, and it readily adds hydrogen to form a dihydride. And so what we're looking at here is the addition of parahydrogen to this iridium complex, and the 2 protons from parahydrogen then bind to the metal. And since they now sit trans to different ligands, so trans to CO or to chlorine, they have different chemical shifts in the product molecule that breaks its symmetry and reveals the hyperpolarization. So in this reaction monitoring experiment, we add parahydrogen to the sample as a gas. So the solution contains our Vasquez complex. Then we add parahydrogen. We shake the tube to put the parahydrogen into solution, transfer it into the magnet, and then over a period of time, we just pulse and acquire on the benchtop NMR spectrometer and observe the hyperpolarized result. And so what you can see is a series of acquisitions here, and these will turn up in the negative part of the proton spectrum, so the hydride region. And so you get a pair because of these 2 hydrides, and they're a triplet because they're coupling to the phosphorus here. But they also have this sort of antiphase behavior that I showed you schematically previously. And what we're seeing here is the reduction of hyperpolarization as we consume this starting material, because each time we just see the hyperpolarization that's been formed since the last observation. And so in order to analyze these kinetics, we can just integrate it, fit our kinetic model, and we obtain a rate constant here. And this is a very well-studied reaction, so we can compare with the literature and see that we're in very good agreement. And so because the parahydrogen hyperpolarization is coming from the reaction, it gives us a way to monitor the reaction in a very robust and quantitative way. So this is a well-established sort of procedure for looking at hyperpolarization, parahydrogen hyperpolarization at high field, but we're just showing here that it's equally applicable on the benchtop NMR, and really gives access to a level of signal that would not be available on the benchtop thermally. So one of the other benefits of using a benchtop NMR is that it's very compact, but we also have a lot of access to the bore of the spectrometer. So the bore of the spectrometer just runs all the way through. And so we are able to couple a UV lamp just through a fiber optic, irradiating the sample from below. And that allows us to look at reactivity with parahydrogen that's initiated photochemically. And so in this example, what we're doing in this first one here is, this is a ruthenium tris carbonyl complex. When we irradiate for a few seconds with the UV, we reductively eliminate one of the carbonyls, creating a reactive intermediate that reacts with the parahydrogen in solution. Again, we've just added it as a gas and shaken the sample to put the parahydrogen into solution. So it adds to form a ruthenium dihydride complex. And again, we see these antiphase, very highly enhanced peaks in the hydride region of the benchtop NMR spectrum. And so we can use this to detect the formation of this ruthenium complex. Now, the hydrogen addition here is very, very fast on the NMR timescale. So it's been measured with other methods to be sort of in the pico to nanosecond timescale. So here we're just seeing the product, and the products are all essentially forming instantaneously on the NMR timescale. However, we can use this initiation idea to look at kinetic reactions. So this is the same reaction I showed previously, but now instead of starting from Vasquez complex, we start from the dihydride complex. When we irradiate, that reductively eliminates H2, creating Vasquez complex, which then reacts with the parahydrogen in solution to reform the starting material, but now with this hyperpolarized peaks. And so we can irradiate and then follow the reactivity over the subsequent minutes to get the same kind of reaction profiles as we saw with the thermally initiated reactions, where we start from Vasquez complex. We can also look at some other more fast relaxing species. So initially we were doing the triggering of the UV lamp manually, which took a better part of a second, but now we've integrated it with just a simple relay, Arduino controlled relay, and that's allowed us to look at hyperpolarization of a different complex here. This is a ruthenium dihydride with 2 bisphosphonating phosphines. Again, same procedure, we irradiate in situ to create the reactive intermediate by kicking up the H2, reacts with parahydrogen solution to reform the starting material, but now hyperpolarized. And what we saw in this case was, with the manual trigger, we didn't see any hyperpolarization, and that's just because the lifetime of the hyperpolarization is quite short. And so we need to do the rapid automatic trigger in order to capture the hyperpolarization. And indeed, we could do a sort of pump probe experiment here, where we vary the time between the irradiation and the detection, and we can see the relaxation of that hyperpolarized response over the course of a second. So this is quite a much more fast relaxing hyperpolarization than the examples in the previous slide. So hopefully what I've been able to show with these hydrogen to PHIP results is that we can fairly easily translate the methods for doing parahydrogen hyperpolarization from high field to a benchtop by just introducing parahydrogen into our sample. And that therefore we can use the well-established tools for monitoring hydrogenation reactions on a benchtop NMR spectrometer, and that by integrating photoirradiation, we can look at a wider range of reactivity. But one of the real challenges to using this form of PHIP, which we might call hydrogenated PHIP, where we're actually using the parahydrogen in a reaction and then we're chemically changing our molecule and looking at the protons from parahydrogen directly, is that it's inherently a one-shot approach. Once you've done the reaction, you have a product material that then can't be repolarized. You need suitable precursors that can be hydrogenated. And so therefore, it's not really a broadly applicable approach if you're thinking about trying to hyperpolarize a wide range of molecules for example, analytical applications. So this is where the method signal amplification by reversible exchange, or SABRE, comes in. So this was an idea first presented in 2009 by my colleague Simon Duckett here at York. And the idea here was to replace that hydrogenation step by a reversible reaction that would allow us to repolarize the same sample multiple times. So I'm going to start just with a rather cartoon view of how this works and then build up kind of some complexity to hopefully bring the understanding of what's happening. So here what we have is, again, we're looking at a transition metal complex like the ones we've seen in the previous slides that binds parahydrogen, form a dihydride. But importantly, it's binding that reversibly. And it also binds other substrate molecules. So this is our sort of target analyte we're going to hyperpolarize. And those are also bound reversibly. And so we start out with a solution of the catalyst, so the metal complex, and our substrate. So that could be something typically like pyridine. We add parahydrogen as a gas, and we shake the tube to put the gas into solution. So our parahydrogen adds to the metal complex. We break the symmetry between the two protons, and we create hyperpolarization on the hydrides. And importantly, if we do this in a very specific low magnetic field of about 6.5 millitesla, then because of the J coupling network across this complex, we get spontaneous flow of polarization from the hydrides to the target nuclei that are bound to the metal center. And then through reversible exchange, we exchange the hyperpolarized substrate molecules into solution, and we refresh the hyperpolarization on the hydride. And so what we establish is essentially a catalytic method for transferring the hyperpolarization from the parahydrogen to a target analyte using this metal complex. And so if we continue to shake our sample and provide fresh parahydrogen over a period of, say, 10 seconds in this weak magnetic field, we build up hyperpolarization on our target analyte in solution, and then we put it into the magnet and we detect our response. And what we get are signals that are enhanced by factors of up to thousands. Typically, these appear as emissive peaks, so peaks pointing down in our spectrum. And this is all coming from this catalytic transfer polarization from parahydrogen. So there are many steps to this and many different factors coming into it, but actually running these experiments are quite straightforward. So just to illustrate that, I just have one of my favorite videos of a former PhD student, Olga Semenova, doing one of these experiments in the lab on a high field NMR spectrometer. So what you'll see is she's got here a tube containing the solution with the catalyst and the target substrate that's been filled with parahydrogen gas. She's shaking that tube in the fringe field of the magnet at a place where we've measured it's about 6 millitesla. So doing that about 10 seconds to build up the hyperpolarization. Then we drop the tube into the magnet and press go, and then we see this strongly enhanced NMR response. So that's our hyperpolarization. So what's really particularly interesting here is that the hyperpolarization, the sort of magic of SABRE, is happening outside of the high field magnet. So you don't actually need the high field magnet to achieve the hyperpolarization. And so that makes it very attractive for combining with benchtop NMR. So what are some of the benefits of SABRE and the features of SABRE? So first of all, compared to the other PHIP methods that I showed, it doesn't require hydrogenation. So it's not chemically changing the target analyte that's being hyperpolarized, which means it's reversible. So we can repolarize the same sample with the addition of fresh parahydrogen. And we get this spontaneous transfer of polarization from the parahydrogen to the substrate or the target molecule via the J coupling network in a weak magnetic field. So it can be coupled with whatever detection field that we want to use. Now, in order for something to be hyperpolarized by SABRE, it needs to bind reversibly to the catalyst on a suitable timescale. It needs to form a suitable J coupling network to the hydrides in this complex to receive the polarization. And it needs to, whatever hyperpolarization we generate, needs to last long enough. So it needs to have a sufficiently long relaxation time so that we can build it up in solution and detect it. So when you watch the SABRE experiment, it does look a little bit like magic that the signal comes out of nowhere, but just to give you a bit of an idea of what's actually happening here, one of the best ways to try and understand polarization transfer in SABRE is to think about something called a level anti-crossing. And this is sort of a view of SABRE that was first introduced by Ivanov a number of years ago, and there's some good reviews on this. So I'm drawing a lot on that in this sort of demonstration here. And so if we just simplify our transition metal complex, so this is the sort of SABRE complex that's doing the transfer of polarization, to just the two protons that have come from parahydrogen, and then one proton on our target molecule that's also bound to the metal center. We can think about our two protons from parahydrogen. At the moment they've added, they're still all in the singlet state. So we've just populated this singlet state. Whereas in a weak magnetic field, our proton that's on the target nucleus is essentially non-polarized. It's a very weak magnetic field, so we would essentially have a similar amount of alpha and beta in this system. And so if we combine these three spins together into one spin system, we can combine all of these states of the hydrides with the state of the target nucleus, and we can divide it into different substates according to the total sort of projection of the angular momentum. And the key point about these sort of substates is just that we can move within each of these groups quite easily, but we can't move between groups. So what we want to do is just focus on one of those. And if we think about our starting point, where starting point are these two states where we've got about an equal amount of alpha and beta, and all of the hydrides is in the singlet state. So if we choose one of these to focus on here, what we can see is what these energy levels are doing as a function of magnetic field. And we're looking at a really low magnetic field here. And what we find is if you set the couplings to 0, so there's no coupling between the hydrides and this spin, then we get the energy levels cross, but there's no interaction between the energy levels. But as soon as you introduce a difference in coupling between the two hydrides in this third spin, for example, if we set this trans coupling to 1 Hertz, then what we see is an avoided crossing at about 6 millitesla. And the important part about that level anti-crossing or avoided crossing is that in the region of this, so in this circular area, that's where we can transfer polarization quite easily between the states. And so how does that work to our benefit? Well, if we start out, as we said, in the singlet alpha state, and we bring it to our sample to the right field, so we bring it to the region of this level anti-crossing, then we can transfer it to this other state, which is the T1 plus beta state. And so now if you think about this spin here, B is now fully in the beta state. And so it has been hyperpolarized. And what you would expect then is to see an enhanced emission peak because you've hyperpolarized the upper spin state. And so that's why in SABRE, we typically see these large enhanced emission peaks that are pointing down. So in order to do our SABRE experiment, what we need to do is we need to work out where this level anti-crossing is, and then we need to carry out the exchange reaction in that field. And so we can work this out by just taking the difference, tweaking that field so that the difference in chemical shift between the two, between the hydrides and the target nucleus is about equal to the effective J coupling of the system, which is typically this proton-proton coupling about 8 to 10 Hertz. And so if you want to transfer to proton, you need a field of about 6.5 millitesla for standard samples. But if you want to transfer to other types of nuclei, you need a much smaller field to bring those energy levels closer together. And so, for example, for fluorine, it would be on the order of microtesla. So a few microtesla. So that's actually weaker than the Earth's magnetic field. But the key point here is that the polarization transfer is spontaneous, so we don't need to do anything for it to occur. We just need to carry out this reaction in the correct field. And so to apply this to a benchtop, all we really need is we need our NMR tube with our solution with the catalyst and the substrate and adding parahydrogen as a gas. And then we need to shake that, so to mix the sample in an appropriate field. So standard benchtop NMRs, unlike high field NMRs, don't have a big fringe field, so there's not much of a field outside of the casing of the spectrometer. So we've just created a sort of handheld magnet that allows us to do the SABRE experiment. This is what our catalyst actually looks like. So it's an iridium catalyst. It binds 3 molecules of our analyte and then the parahydrogen. And what you can see here is that on our benchtop, we get huge enhancements because we're starting from such a low level of signal for this very simple molecule here of methylpyridine. And we can see both the enhancement of the methylpyridine peaks in free solution and also bound to the catalyst. And one of the benefits of the benchtop is that we can actually use protonated solvents, and you can see the hyperpolarization overwhelms the protonated solvent response. Another challenge of benchtop NMR is that even for these very simple molecules, so this is a range of fluoropyridines I'm showing here, you get a lot of peak overlap because of the compaction of the chemical shift axis in low fields. So we might think about using other nuclei to detect. So something like fluorine is very analytically interesting. It's in a lot of things like pharmaceuticals, has a wide chemical shift range, and will have minimal background signals. But for SABRE hyperpolarization, typically we get less hyperpolarization on the fluorine. So if we're looking at doing fluorine-enhanced benchtop NMR, there's additional tricks that we can use to further boost our signal to noise, because we're interested in trying to see how low can we take our limits of detection using SABRE enhancement. So I just want to mention, just because it comes up in some of the results later, one of the tricks we use to further enhance the signal to noise of our fluorine signal, and that is this sharper pulse sequence, which is just about collapsing multiplets into a single high SNR peak using a sort of CPMG-like train where we acquire our FID between all of the pulses. So basically how this works is we excite our signal, we acquire a small piece of our FID, and then we do an echo train to refocus chemical shift, field inhomogeneity, and J coupling, and acquire our pieces of the FID and stitch them together. And then we get a narrow peak that has a much higher signal to noise. And we can combine this with fluorine enhanced SABRE on the benchtop to basically increase our signal to noise by, on average, about a factor of 10 beyond what we already get with the SABRE. So we have this combined effect. So now that we have all of these tools, we want to look at how we can actually use them to improve the limits of detection of the benchtop NMR. But we're also very interested in whether we can quantify the signals we get. So one of the big benefits of NMR in general in analytical applications is that it is relatively easy to quantify. But you give that up to some sense when you're using hyperpolarization, because the amount of signal you get depends on how efficient your hyperpolarization is, as well as how much of the molecules in your solution. And so you need to have really consistent hyperpolarization, or you need a way of calibrating that so that you can relate the signal you get to the amount of substrate or molecule of that molecule that is in your sample. So in order to try and look at quantification and limits of detection, we're going to add one more thing into our SABRE process, and that is the use of something called a co-substrate. So normally our standard SABRE catalyst looks something like this. We have a iridium center. There's a large carbene here. So this is a sort of bulky molecule that sort of stabilizes and creates the final catalyst. And we have the dihydrides that come from parahydrogen. And normally this molecule is activated using 3 molecules of the target analyte. But that's not desirable in many cases, in particular if we're trying to look at things in solution that are present in only very, very low quantities. And so the idea of using a co-substrate is you add something else into your sample that can create this active catalyst. So you kind of tweak the active catalyst. So some of the sites are occupied by this other molecule, and there's only one site free for the target molecule. And this has been shown to help expand the rate, the scope of analytes can be analyzed with SABRE. So there are a number of molecules that are weaker binding, or they're hindered in some way from binding to the catalyst. And so by using co-substrates, for example, my colleague Simon Duckett has shown that we can hyperpolarize pyruvate, which actually binds the catalyst in a bidentate way, so through two oxygens here. And so it was through tweaking that co-substrate that enabled that to work. And very interestingly, as well, the group of Marco Tessari has shown that by using this co-substrate approach, that they can actually achieve quantification in the very, very low concentration range. So down at a few micromolar here through standard addition, they're able to quantify the components in a very complex mixture, with the idea that the whole suite of potential analytes for the catalyst are acting as co-substrates. So to test this on the benchtop, we're using the same test molecule as in some of the previous results I showed, which is difluoropyridine. So pyridine we know binds very nicely to our catalyst, and then we have the fluorine for looking at the fluorine NMR. And here we're mixing this with DMSO as a co-substrate and our standard our iridium catalyst. And so the main species in solution has two molecules of DMSO bound to the iridium, but some of the iridium molecules also have instead the difluoropyridine attached, and these are the ones that are hyperpolarizing our difluoropyridine. And just showing here that using this mixture, we can get hyperpolarization on both the proton and the fluorine for this lower concentration. So just looking to see now with this sample how low we can go in terms of limits of detection. First, starting just with the reference point. So if you take difluoropyridine and methanol, if we go down to about 8 millimolar and we signal average for about 15 minutes, we get signal to noise of around 7. So that's very much our limit of detection using standard benchtop NMR at 1 Tesla. Now, if we use SABRE, what we find is that in a single scan, we can reduce our concentration of difluoropyridine down to about 14 micromolar and get a decent signal to noise ratio. And so for the single scan limit of detection, that's an improvement by about 4,500. And we can push this even further by using this sharper sequence that I explained. It gives us an extra 12-fold SNR enhancement, and we can exploit that to detect difluoropyridine down into the hundreds of nanomolar range. So really, by putting all these things together, we can really start to detect very low concentrations, even on a 1 Tesla benchtop NMR spectrometer. And finally, just looking at quantification, what we find is in this 100 of micromolar range of difluoropyridine, again in the same sample using DMSO as a co-substrate, we can see that we get a nice linear correlation between signal and concentration of difluoropyridine in for proton NMR, and a test sample gives a recovery of 107%, so very, very close to the ideal of 100%. We have very good linearity. If we look at 19F SABRE, then we get, again, very good linearity and very good recovery for our test sample. And this is improved even further when we look at the SABRE sharper, because we've increased our signal to noise by the sharper, so we get better linearity and excellent recovery. So this is just looking at a single component in our sample, and so in order to broaden this out, we need to explore what happens when we have multiple components in a mixture. But as shown at high field, we can also address that using standard addition, but we want to look at the other ways of doing that as well going forward. But it shows a very promising route to quantification with SABRE and benchtop NMR. So finally, just to finish on the sort of SABRE enhanced benchtop NMR, can hopefully shown you that we can integrate this in a quite straightforward way and enable low micromolar limits of detection. And that by using this co-substrate approach that we can actually access quantification as well. But that there's still more work to be done on looking at interference and complex mixtures. And also the experiments I'm showing were all done manually using the shaking approach, but we can also automate this using bubbling of parahydrogen through the sample. And also we're looking into methods for doing in situ SABRE transfer, which will have many benefits as well. So just to finish with, thank you to my research group and to various collaborators and funders over the years, and thank you for your attention. I'm happy to answer any questions.
Thank you very much, Meghan, for this wonderful tutorial. There is already a question from Nasreen. So the question is PHIP sample specific or is it only nuclei specific? If the sample has, for example, hydrogen and fluorine, which field will give higher polarization?
So in principle, the ideal, like the actual perfect PTF will depend on the exact sort of spin system. So it depend on the exact catalyst you're using and the exact molecule that you're targeting. But what we find in practice is that these sort of resonance conditions are quite broad. So for because the sort of ppm difference between the hydride, so the protons that come from parahydrogen, and the organic region is pretty consistent between a lot of target molecules, that there's a good compromise field that gives good hyperpolarization for most things, but it's something that can be tweaked in the development. Now, if you're looking at transfer to fluorine, so if you're doing direct transfer, then you do need a much weaker field, and it's, as I said, it's sort of below the Earth's field, so you need to create some kind of shield to shield the sample from the external fields in order to get transfer. But actually what you find is that you can, and in a lot of the experiments I showed, we were actually doing the fluorine experiments at 6 millitesla, and what's happening there is you're first essentially hyperpolarizing the protons, and then the proton is mixing with the fluorine and transferring that polarization to the fluorine. And so in general, you can usually get pretty efficient hyperpolarization using just that standard 6 millitesla field for a lot of target analytes.
Mm-hmm. I hope that answers your question, Nasreen. There is one which is regarding parahydrogen, shaking the sample. So the question is, shaking sample outside a 400 megahertz magnet prefers over an unshielded magnet. Can you confirm this?
Yeah, so in for the ones where you're using a fringe field of the magnet, you would use a Gauss meter to try and locate approximately where the 6 millitesla is. So that's quite and then you shake in that region and then put the sample into the magnet. So that's quite convenient, but it's also very irreproducible, and then it will be very different, like where it is for a shielded magnet and unshielded magnet. So actually in practice, it's fairly easy to make a handhold magnet from permanent magnets at the right field of a few millitesla. And so I would say that most experiments done in the lab, even when you're using high field detection where there is a fringe field, are done inside a holder where, like a handheld magnet that's creating the field that you want. So you get a much more homogeneous and consistent field rather than use the fringe field. So the fringe field is convenient if you're just doing some one-off measurements, but it's better to do it in a specific field where you know what that field is by using a handheld magnet.
Just a curious question. So after you shake the sample and putting it in the magnet, the magnetic field is changing in the process. Does that, in some sense, it's a sweep? So does it have an impact?
Yeah, so we definitely see some evidence that potentially passing through those different fields can accomplish some transfer. So in general, because of the time scale, the time it spends moving through those fields is not very long. So there's not usually that much transfer, and that the bulk of the transfer is happening during sort of the 10 seconds that you're shaking in the desired polarization transfer field. So that's pretty much for hydrogen. We're pretty confident that most of the transfer is happening outside in the target field, because as it goes through, particularly the bore of the high field magnet, it's just going through higher fields where it's very much out of resonance, so it's not going to create transfer. However, we do think there's potentially some effects that we're seeing, for example, on the fluorine, that we're shaking in the 6 millitesla, which in principle is hyperpolarizing the protons, and that by taking it out of the shaker and moving it through into the spectrometer, we're passing through a number of fields, and that may be aiding the mixing of the hyperpolarization from the proton to the fluorine. And so that's a bit harder to control in these types of experiments, but we think it may be helping there in the fluorine measurements.
Okay, thank you for that answer. There is a question by MJ. He thanks you for the talk, and his question is, have you had the chance to test PureShift NMR pulse sequence for other nuclei as well, such as phosphorus and oxygen?
Yeah, so the sharper pulse sequence that if that's what you're referring to, that I showed, we have done that on phosphorus as well. It works really nicely. In fact, we've done some this collaboration with Leibniz that I've got on here. Laura here, who's in the middle of this picture, came and visited us last summer, and she's an inorganic chemist, very interested in looking at catalysts, especially with phosphine ligands, and trying to detect them and reactivity involving them on a benchtop NMR. And so we implemented this sort of sharper method to boost the SNR on phosphorus quite successfully, to be able to look at faster reactivity with less signal averaging. So that was not hyperpolarized, just with the sharper method. There are, of course, lots of different types of PureShift methods, and if we're talking about PureShift in the sense of like in proton, where you're trying to remove the homonuclear couplings, that's something we know has been done with SABRE at high fields, coupling PureShift like Psyche with SABRE, and we'd be very interested in we're looking into testing that out on the benchtop as well, but haven't done that yet.
There is a similar question, which is, have you tested quantification of a more complex mixture, and if there are any results on that, and also if you have been able to solve the problem of resolution?
Yeah, so in terms of complex mixtures, so like I said, it's been demonstrated at high field that if you've got a complex mixture, that you can use standard addition, so basically spike your sample with the target and then back extrapolate to the amount that is in the original mixture. So that basically allows you to test to extract that sort of coefficient of the efficiency of the hyperpolarization in the actual matrix that you're looking at, if that makes sense. Because the issue with the SABRE quantification is that when we see this nice straight line, if I can go back, these nice straight lines, the slope of this line tells us something about the SABRE efficiency, and that SABRE efficiency is going to be really sensitive to sort of competition for the catalyst. So if there's lots of other things in the mixture, the competition will change, and so that slope might change. And so that's why doing the spiking of the sample, the standard addition, allows you to extract the concentration within that complex mixture. So what we are hoping to try and think about is how you could develop a different kind of method using something that acts more like an internal standard, so that we don't have to do the standard addition. But that's very much at ideas and development stage. But we do know that as soon as you add in a competing ligand, there's the chance that this calibration curve will change. and so it'll affect the quantification. You'll still get a linear response, but the slope will be different depending on what else is in the solution.
Moving on, there is a question regarding biological application. So someone mentioned Edward. He says for biological substrate, it's important that the sample is purified from the catalyst, and he wants to know what's the general purification procedure.
Yeah, so a lot of people are working on SABRE to generate so hyperpolarized contrast agents for example, for in vivo MRI. So there, where you're really thinking about injecting it into a person, both, you know, there are many things you have to think about. You have to think about the solvent. So everything I've done here is in methanol. Obviously, that's not something that you would inject. So a lot of people looking at sort of ethanol mixtures. Then and water. So water is a bit of an issue, just because hydrogen is not very soluble in water. The catalyst doesn't work as well in water, and so on. So some of the potential ways that this can be done, has been demonstrated to be done, is using a biphasic approach. So basically, where you have where after you have an organic solvent where the catalyst is, and then a water layer, which is where your substrate can be in both layers, so it gets hyperpolarized in the organic layer, and then you can extract the water layer, which then would not have the catalyst in it because the catalyst is not soluble in water. There's also methods where you can basically kind of put a scavenger in to sort of remove the catalyst. And there are a few methods and few demonstrations now that we've been able to hyperpolarize pyruvate. There's some examples in the literature of people then purifying those samples using a combination of those types of methods to try and remove the catalyst and get a pure hyperpolarized substrate. But the problem is that once you create the hyperpolarization, it starts to relax. So all of those things need to be done as quickly as possible and as efficiently as possible, so that you don't lose all of your hyperpolarization. So typically for those kind of things, you would hyperpolarize carbon-13 because it has a much longer relaxation time, so that then you have maybe a minute relaxation time to do all of those steps.
This is just out of my curiosity. I've seen the company Envision getting a polarizer for pyruvate. Do you can you provide some insight about their setup?
Yeah, so I think I'm right in saying that what they're doing is something called PHIP-SAR, so side arm hydrogenation. So what they do is they take the target molecule that they want to hyperpolarize, so for something, for example, the pyruvate, and then they synthetically add a side arm that has a double bond that can be hydrogenated. So they hydrogenate that part of the molecule, transfer the polarization to the bit of the molecule they're interested in, and then cleave it chemically, and then do the purification and inject. And so and a lot of the same considerations are there about how they separate it out. I can't remember off the top of my head exactly how. I think they dilute with water, and then either they rapidly precipitate and then redissolve it. I can't exactly remember how they're doing the purification, but I think the work that they've been doing is using this sort of sidearm hydrogenation approach, which is a very cool chemical method for getting around the problems of hydrogenative PHIP and getting to the target analyte that you want hyperpolarized.
And in scenario where there are multiple protons, how does the polarization transfer to the other protons in the molecule?
So essentially in the low fields, because the field's so small that the difference in chemical shift it becomes almost negligible. So here you've got two protons ortho to the nitrogen and then the paraproton. And so once you're in this very low field of 6 millitesla, these are very similar chemical shifts. So essentially their frequencies are aligned. And so you can get mixing of the polarization between them quite efficiently. And so here what you're seeing is, I believe this are the orthoprotons, and here these are that's the paraproton. so that's coupled to the fluorine, so that's why it's split or broader in this example. So you will get different levels of polarization, but essentially the polarization kind of distributes throughout the molecule in the same way that, here it's also distributing to the fluorine, and we're detecting the fluorine.
So this is always through J coupling exchange.
Yeah. So there's some work looking at to see if there's any contribution from like cross relaxation and that kind of thing. And it was established fairly early on in SABRE that the main component of the enhancement that you see in this sort of approach, where you're working at 6 millitesla and doing the shaking, is through sort of coherent transfer through the J coupling network.
Mm-hmm. We are running out of time, but we'll take 2 more questions, which are already there. One is by Victor. He wants to know about hydrogen gas generator. Is it convenient to use? Is the hydrogen gas pure enough, or are there some issues with the flow rate?
Yeah, so the hydrogen gas, the most important thing with the hydrogen gas generator is that the hydrogen that comes out is dry, because you're then going to cool it down. And if it's got any impurities or it has water in it, then that will freeze and then block your parahydrogen generator, the coil. And so that's just about making sure you're getting the right grade of hydrogen generator, and making sure that there's a good filter unit on it so that it's pure, because basically that's really the problem, is when you're cooling it down, if there's anything in there that's going to freeze, it'll block your system up, and the hydrogen won't be able to flow through anymore.
Mm-hmm. And the one very last question is by Gabriel. He wants to know, when you apply SABRE sharper technique, you have to shake your sample at 6.5 millitesla, and then they apply the pulse sequence at higher field at 1 Tesla. So his question is this an approach that can be used for any nucleus in particular, in principle?
Yeah, so it can be used for any nucleus. So, I mean, you're removing all of the chemical shift information, so you're collapsing everything in. So it's particularly useful for like fluorine or phosphorus, where you only maybe have 1 signal or 2 signals. So you can selectively excite a group of signals to collapse as well. But if you've got, like in a proton spectrum where you've got lots of overlapping peaks, then the sharper is not going to be useful, because it's just going to collapse everything together, which is maybe not the signals that you want. So what we are looking at in terms of proton is looking at some other sequences where you can do effective selective excitation. So things like there's a sequence gemstone that does sort of selective excitation of overlap peaks, because then once you did that, then you could in principle collapse those down to increase the signal to noise. But it's most useful in its sort of standard form for heteronuclei, where there's not a lot of peaks, and you're not looking for that chemical separation necessarily.
Mm-hmm. This is the question that just came to my mind. So suppose someone wants to enter into this research, what would be the cost of parahydrogen generator if you are doing home build versus buying a commercial one?
Yeah, so I'm not sure. So there's a number of people now who I think are selling nitrogen-based commercial ones, which, and I mean, I've never bought one myself, so I don't know, but but that will be certainly much cheaper than, say, the Brooker one, which is operating at a much lower temperature. There's sort of a minimal generator that you can make, which is that doesn't have a large reservoir of nitrogen. So if we go back, if I can get to, it's going to go through a lot of slides. So I think early days, there were some publications where there was suggestion that you could get all the parts for a few £100 or a few $100. I think it's possibly difficult to do that if you're like, we built in lots of sort of safety features into the swage lock here, and swage lock can be a bit expensive. So there's a very simple way where you're just having the hydrogen flow through to an outlet. Here we're using a large dewar, which is of course expensive, but that's just so that we can run for a long period of time. You can use a much smaller dewar. So it's not particularly expensive. There are a number of designs in the literature, but it does take a little bit of know-how, how to sort of pack the coil with the iron oxide efficiently, and set up the valve so that you've got, you know, some pressure regulators, and make sure you've got everything operating safely. The other thing that's not pictured here is that you need a vacuum pump, which again can cost a bit of money, but yeah, I guess it depends on how complicated a setup that you want, but there are a number of different sort of cost-effective designs in the literature. These days, with the increase of cost of everything, I'm not sure if you can do it under £1,000 now, but you certainly used to be able to do it for a few hundred pounds for the very, very basic setup.